We welcome you all to the MTX Day 3 of Shaastra 2021 and the next lecture of Spotlight. Keeping up with our aim of inspiring, energizing, and motivating the student community, up next in our star-studded lineup, we have with us today Dr. Joachim Frank, Nobel Laureate in Chemistry and founder of the Cryo-Electron Microscope. Dr. Frank is a professor in the Department of Biochemistry and Molecular Biophysics at Columbia University, with his lab known for developing path-breaking techniques of single-particle reconstruction of biological macromolecules, specializing in mathematical and computational approaches. Dr. Frank has made significant contributions to the structure and function of ribosomes from bacteria and eukaryotes. He shared the 2017 Nobel Prize in Chemistry for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. We are very honored to have you with us, sir. So without any further ado, we would like to start off with your lecture. So I hand it over to you.
Thank you very much for having me here. I enjoy being part of this conversation. So I'd like to tell you about single particle cryo-EM, deciphering the building blocks of life. I was actually in India just the month after my Nobel Prize was announced, because the arrangements were done much earlier, and I was in Varanasi in November 2017. So we are asking, what is life? We see life in these incredible variable forms, and we want to know the basis of it. And we can make discoveries as we zoom in on parts of an organism with increasing magnification. There's the organism, and then there's the level of organ, tissue, cell, organelle, and then at the smallest level is the molecule. The smallest level of life is the molecule itself, the molecules of proteins and RNA. So we have discovered in the process that the life functions are increasingly looking the same as we go to higher and higher magnification. So the basis of all forms of life is pretty much the same. And here's an example for an animal cell. And you can see many compartments, many different organelles. And but to know how everything works together, we have to get on the molecular level. And so I repeat, to understand life, we need to understand the structure of molecules and the way they interact. And we have an important concept, which is the concept of molecular machines, that was popularized by Bruce Alberts in 1998. Many molecules act in concert in a processive way. And now it is very difficult to look at the molecules at the highest resolution level in the context of the cell. So we employ what we call reductionism. We study a subsystem in isolation in vitro, and we hope to approximate the processes in the environment of the cell. And here are examples of molecular machines. ATP synthase makes ATP, proteasome recycles proteins, RNA polymerase copies DNA, and the mRNA ribosome makes proteins. That's my favorite molecule because it's been used to develop the mathematical and computational techniques. The chaperone folds proteins, the dynein transports molecules, the spliceosomes edits mRNA, flagella motor rotates flagella. These are just a few examples of hundreds of molecular machines that all are in our body. So there's been a technique that has been long-standing and has been used for decades, which is Xray crystallography. In a crystal, many copies of a molecule are arranged in regular order. And when we expose a crystal to the X-ray beam, we get a diffraction pattern. And then by using some methods of phase determination, we get a structure. And the heroes here, the pioneers, were Max Perutz and John Kendrew, who got the Nobel Prize in chemistry for determining the structure of hemoglobin. This was at the time the largest molecule that has been looked at by X-ray. The X-ray beam must be high intensity for proteins, and the crystal must be almost perfect. And today, more than 140,000 structures are solved by Xray crystallography, and they're all available in the public data banks. And they are basis of what we call the molecular medicine. But because of the crystal packing, molecules cannot be visualized in all conformation and binding states that are important for function. And what's more important, many molecules do not form highly ordered crystals, so they're completely absent from the public database. The sample quantity can be a big issue as well. So this brings us to the use of the electron microscope in order to directly visualize molecules. Now, what is an electron microscope versus a light microscope? Well, it's a microscope that uses electrons instead of photons, and you can look at the two apparatuses in strict equivalence. You have a lens instead of a glass lens, you have a magnetic lens. The very big difference is that electrons need to be in a vacuum to travel, because otherwise they would be absorbed very quickly in the air. And the huge difference is that the light microscope has a resolution limit of the wavelength, which is 400 nanometers. There are some super resolution light microscopes, but they still don't go much farther than this. Electrons go to less than 0.1 nanometers or 1 angstroms, and so they are perfect in visualizing such structures. So the electron microscope itself is a very large instrument, which is the size of a room or even larger. So as I will show, it can be used to solve molecular structures as well. And projection images are formed with high magnification. For instance, typically is 30,000. To reconstruct an object, many different views must be collected. The sample must be very thin, though, because electrons are readily absorbed by matter. And a big problem is that electrons strongly damage the molecules. And this means that there's a need for low dose. 10 to 20 electrons per square angstrom is sort of the level that is used. And as a consequence, the images are very noisy. We talk about shot noise. And as a consequence of this, we really need to average. We need to average over many images in order to make sense of the image. And then initially, negative staining needed to be used for sample preparation. So I did not develop my computational methods using cryo methods. They came out later. So that I want to emphasize. So here is a transmission electron microscope. We have a high voltage source, the electron gun, and between cathode and anode, electrons are accelerated to a very high velocity, to 100 kilovolt, 200 kilovolt, 300 kilovolt, and so forth. And then they travel through an illumination system, and the sample is in the immediate vicinity of the objective lens. And that's the important lens which forms an image. And that image is then magnified in 2 stages. And in the end, it used to be observed directly on a fluorescent screen or recorded on a plate camera. And nowadays, there is an electronic camera right here. Okay, so negative staining, the principle is that the molecule is embedded in a heavy metal stain salt. So we see the molecule only because we don't see it. We see it as a stain exclusion. And this has, they have high contrast, but the interior of the molecule is not seen very well. And that's why the introduction of cryo methods later brought a real revolution. And initially, people looked at crystalline arrangements of molecules. That was essentially a thinking that they had with X-ray crystallography. They thought, you know, well, if we want to use the electron microscope, then we also need to have molecules in highly ordered arrangements. And these are examples. They are all from the Medical Research Council. Helical structures, icosahedral structures like in viruses, planar structures. And this then leads to the question, you know, why do we need crystals? Because if we need crystals, then we limit our whole variety of molecules to a very small subset. And so my mentor, Walter Hoppe, was asking a question why we need crystals at all, and so he thought of tilting the molecules, having the molecules on the grid, and then tilting them in even increments to high angles. And from the tilt series, one can then reconstruct the molecules as they are on the grid. And the problem with this is that we have an accumulation of electron exposure which exceeds 1,000 electrons per angstrom square. And under those conditions, the molecules simply burn up and we get a reconstruction, but the reconstruction is meaningless. Okay, so my idea was to circumvent this entirely by thinking of using molecules that are simply free in solution, and they are then air dried and negatively stained as the sample preparation was at the time. But the whole idea is really to collect structural information from single images of single unattached molecules that exist in many copies, and they are free to assume all naturally occurring conformations. They're randomly oriented, and a single snapshot may already give us hundreds of particle views. And if 100's not enough, then we collect more and more and more, and enough to fill the entire angular range. So this is all a grand idea, but the devil is in the details. One has to develop methods for all the different subsets of techniques. As we need to align images, we have to sort to classify them, we have to find the projection angles. That's the most important thing, because we don't know the projection angles. The molecules have simply have sought their own angles, and then reconstruct them with generalized geometry in 3 dimensions. I have only indicated the various problems here in these little insets. And the important thing is that I needed to make myself a workbench of programs in order to really deal with all the hundreds of problems that needed to be solved. Okay, so I cannot go into the details, but I managed to do this essentially within 10 years, from 1975 to 1986. So here is just alignment and averaging. This a proof of concept paper that I published in 1981. Here's one where I addressed the problem of heterogeneities in order to sort out molecules in different orientations and conformations. That was around the same time. Now, there's one particular person that I want to single out, which is Michael Rademacher. He was a postdoc of mine, and he specifically developed the programs for searching for finding orientations and reconstructing them in 3D. So he is sort of a really hero in this story. So this published in 1987, it was the first single particle reconstruction ever. It was a reconstruction of the 50S ribosomal subunit from E. coli ribosome. Here it's shown displayed at different density levels. And this was still negative stain reconstruction. And then there was really this revolutionary introduction of the ice embedding techniques by Jacques Dubochet, who with me received the Nobel Prize later on. He did this in 1981, but the technique only became popular a few years later. And essentially, the sample is put in a droplet on the grid. It contains, thousands, millions of molecules, and then the excess is blotted away so that a very thin water layer only remains. And then this is plunged into liquid nitrogen and into liquid ethane, which is under temperature of liquid nitrogen. And the introduction of liquid ethane here in this process was really the decisive invention that made the whole thing happen. Here is a ribosome as recorded on film. You see that they are very it's like a salt and pepper appearance, which comes from the low dose. And then also the quality, the recording quality of film is really not optimal. So we get these faint images. And so using the cryo technique with a single particle reconstruction methodology gave us an immediate advantage, and we got three-dimensional images of molecules that had never been seen before because they could not be crystallized. And here is a whole gambit of molecules that we immediately were able to reconstruct. Okay, so today's electron microscopes are of this kind of type. And in fact, this is now antiquated and is superseded by a very large box, which is the Titan Krios 300 kV transmission electron microscope. So all the stuff, the good stuff, is inside of this box, and the whole box is climatized. Okay, so we now really have a complete pipeline of biochemical preparation, cryo-EM sample preparation, imaging, data collection, image processing, reconstruction, structural analysis, and then from the reconstruction, we can build atomic models if the resolution is sufficiently large. And as you will see, in fact, we get several reconstructions from the same sample because of the power of today's programs, which are able to extract all the coexisting structures all at once. And this is done with maximum likelihood methods of classification. So we are able to find all the coexisting structures in the same sample all at the same time. And I call this story in a sample. Okay, and then a huge revolution occurred in 2012 that enabled the methods that I had developed to go to atomic resolution. And it was the introduction of a camera which is able to collect every single electron. So it has a much higher signal to noise ratio than film, and it will get us directly at atomic resolution. Here are ribosomes recorded in such a camera. And this here is an example where we get all these different structures from the same sample. And so we get not just one answer, we get several answers to the questions that we asked in the same sample. And this was the first time that we used the technique at very high resolution, 2.5 angstrom, and we see individual ions. We see base pairing in great detail, alpha helical structures. We even can spot individual water molecules. Okay, so here I show you this. I show you this structure just because it's so beautiful, and we can identify the different tRNAs and an elongation factor attached to it. And here, now we have a concept of local resolution. The resolution is different at different parts. What you see in blue here is the highest resolution. It goes all the way to 2.3 angstrom. And at the outside, there's a little floppiness because the molecule is more exposed to the thermal environment. Okay, here's an as we got at this stage, there was a lot of interest by other people to collaborate with me on ion channels, and this is just an example of an ion channel that we solved in recent years. The AMPA receptor, which is important at neurojunctions. And then I had a collaboration on the ryanodine receptor with two groups at Columbia University. We got 3.6 angstroms, and we got the channel in different conformational states. The ryanodine receptor or calcium release channel is everywhere in our muscles, is involved in muscle contraction. And here is an ion channel that is important in cystic fibrosis. This here is a structure that was gotten by Rockefeller Institute, and here are atomic structures that we obtained of a mutant that is important in the disease process. And just as an illustration of where we are, at 2.2 angstrom resolution with beta galactosidase, it was shown for the first time how beautifully we can see the atomic signatures of the various amino acids. So you can sequence simply going through the through the density distribution, and you don't even have to know the sequence of the structure from other investigations. And then that brings me to the coronavirus. We are really fortunate that we had single particle electron microscopy at the onset of it. And in fact, we can account a number of viruses that we have experienced between 2012 and now, and it includes MERS, Corona, Zika, and SARS. So without cryo-EM, we would not have been able to get to structural information that is important and in fact crucial in the development of effective vaccines and antibody treatments. And so this group of Jason McClellan was able to get the structure of the spike protein, which is important for understanding the invasion of the virus into the host. He was able to get it just 2 weeks after receiving the sequence of the virus from the Chinese researchers. That was a year ago. And on the basis of this discovery, it was the drug companies got crucial information in order to proceed with the development of the vaccines. So here is here you have an idea of the numbers of released structures per year. So it's the rate of depositions. You can see that the rate of depositions has leveled in X-ray crystallography, but it's going steeply up in electron microscopy. So since the rate is going up, we get sort of an exponential increase in structures over the next years. And in conclusion, single particle cryo-EM opens a new era in structural biology. There's no need for crystals. Very small sample quantities are needed. Resolution in the 3 to 4 angstrom range is now routinely achievable. And the best resolution so far is 1.5 angstrom. Multiple structures can be retrieved from the same sample, which gives us clues on function. Molecules are enclosed to native conditions. And solving structures of membrane proteins is much easier than with X-ray crystallography. And as I told you, we have experienced a huge expansion of structural database relevant for molecular medicine. Here I just have a few slides that, that recalls for me the glorious days in Stockholm in December of 2017. Here I have the handshake with the king. And here we are actually in a rehearsal of the ceremony, and here are all the other laureates. Here are the 2 others in chemistry, and here I'm presenting to the Nobel Museum a token that is going to be on display. It is a light box that shows transparencies overlapped, which was a primitive way at the time to show 3-dimensional structures. Next to me is Jacques Dubochet. He presents the first plunging device, and Richard Henderson shows the atomic structure of Bacteriodopsin. And here we are in front of the portrait of Alfred Nobel. Here I'm with my wife, Carol Saginaw, who has been incredibly supportive over all these years. And here we are on top of the building in Manhattan a couple of years ago. Thank you very much for your attention.
Thank you so much, sir, for that really wonderful and beautiful lecture. We would now like to proceed with the question and answer sessions. So we would be very glad if you could answer some questions for us. So to begin with, the first question would be, on October the 4th, 2017, at 5:18 New York time, your phone rang. What went through your head at that exact moment?
Well, it was already known that this was going to be the morning of the announcement of the Nobel Prize in Chemistry. So I had a certain inkling because I knew that I'd been nominated a number of times before. And so there was the
Right.
possibility that was the call, and so it was very exciting to hear the telephone, and
Right.
so
Right.
when I then picked it up, and then I heard a woman with Swedish accent talking to me in English. I knew this is going to be it, and she was the secretary of the actual official who who gave me the news.
Right. Okay, that's a really interesting story. Um, so the next question, what were some of the major limitations of the early modes of electron microscopes and imaging techniques, including the early prototypes that were used in 1931, such as Xray crystallography, that began to be utilized in the early 1950s, and the nuclear magnetic resonance in 1980s? In what ways was your discovery innovative and unprecedented?
Well, I touched upon this already in my lecture. With in relation to X-ray crystallography, there's a clear advantage that we can look at molecules that are not that cannot be put into crystal form. So that's a very easy thing to answer. With respect to NMR, NMR has really a limitation in the size of molecules that can be characterized, and so it's really NMR is really good in a resolution range where electron microscopy cannot even reach. Very small molecules cannot be handled with cryo-EM.
Yeah. All right. Okay. Um, how did the Nobel Prize in Chemistry change your life and career? Had you ever expected such a recognition, or like, I mean, as you already said, you were already sort of expecting it, but how did it change your career over time?
Well, it's essentially you get some kind of get an immediate boost in authority. So
Right.
whenever when you open your mouth and people listen, and and
Right.
and it used to be I used to be interrupted more
Mhm.
when I talk. So this is and then obviously it's a great feeling to get a recognition for for lots and lots of work that I did over the decades. Um, and then how it changed my life? Well, obviously, I've been traveling quite a bit, and I've been
Mhm.
able to meet people that I would not have been able to meet uh,
Right.
in conversation. For instance, Ishiro Kazuo Irishugo, the literature Nobel Prize winner, I can chat with him, which is an unbelievable experience.
Right.
Uh, and then what it also changed, I think I'm able to take more risks in projects that I take on, uh,
Mhm.
and also the very fact that I'm still doing research is quite unusual. People at my
Right.
age would normally be in retirement, and that's
Mhm.
probably what I would have chosen without getting the prize. But now I'm sort of encouraged to dream on. Okay.
Right. That just sounds very brilliant. Uh, How does your innovation in cryo-electron microscopy make it possible to carry out detailed observation of the dangerous Zika virus, which was otherwise impossible?
Well, the technique simply gives us atomic information, and on that basis we can then uh, uh, deduce the way the virus interacts with the receptors of the host, and we
Okay.
can design effective drug um, drugs, and so it's a universal technique. So whatever virus comes along, we are able to handle it. Ebola is one of the ones that was on my list before.
Okay.
And I already said that it's very fortuitous that we have this technique now in fighting the coronavirus. Without it, it would have been much harder because Xray crystallography is not able to give us so much information about the conformational changes, and these are very important to know if we want to know how to combat the virus.
Right. Okay. Really appreciate that answer. So, something really interesting. At the same time as being a scientist, you are also a fiction and short story writer. Have your scientific achievements boosted your literary career and brought your works of literature into limelight?
Well, it's yeah, it has helped. There there were certain journalists that somehow focused on the literary achievement. So I got into an interview where by the Washington Post, and all of a sudden found myself in a one page article in the weekend edition of the Washington Post, where only my literary work was featured. And subsequently, I had I was invited by the New York Writers Institute to speak about my short stories and my literary work. So this is very clearly without a Nobel Prize in chemistry, I would not have gotten into that track.
Okay, that's really good to hear.
Yeah.
What's your advice for young aspiring scientists who want to follow your path and make groundbreaking achievements in their fields of study and research?
Well, following somebody's path really doesn't work. The path is completely erratic and idiosyncratic. It is, your own path, and you have to find a way of following your own instincts, and be assertive at the right amount of time. And what I normally say is the key is peripheral vision. You have to take advantage of very unexpected things that come along, uh, and they are always in the periphery. And they are unplanned, and you have to develop an extra sense of perception for those things.
I see. Hmm. So yeah, I hope all the viewers watching this will take note of that. Uh, so to continue, we have some questions by the audience. We have a few questions. So the first one is by Saurav Ghosh. Saurav Ghosh asks, till which level can the protein structures be visualized using cryo-electron microscope? Can we visualize the atoms with this microscope?
Well, we already are at a range of a resolution where individual atoms can be seen. 1.5, in fact, the record is now 1.2 angstrom, and with 1.2 you can clearly see the atoms. And so, but the question is simply, do we need that resolution? We need the resolution that we need is depending on what we want to use the information for. Um, I would say we wouldn't need 1.2 angstrom in order to define drug targets. We don't need that resolution. So it's really the amount of efforts that go into a structure have to be calibrated by what exactly you need. And very often 3 angstroms is sufficient in order to define the atomic structure. And that might be all that you need.
Okay, thank you for that answer, sir. The next question is by Anirudh. Anirudh asks, is there any reason there are so many mutations of the COVID strain? Do you think there is any way to control this?
Well, uh, there, I don't think there's a way to control it. The virus is doing what the virus likes to do, and so it's a very cold struggle for survival, and
Okay.
it takes advantage of a mutation. And if we challenge the virus with a particular vaccine, then there's always there are some survivors, and those survivors have an advantage, and that's why we get such a large number of of mutation. Now, these mutations occur at the crucial place where we have interactions between the spike protein and the receptor, and so it becomes a rat race between vaccine development and the development of mutations of the virus. And it's been pointed out by some people that well, there's a whole metabolism of the virus that can also be attacked. And the RNA polymerase and lots of the uh, proteins that the virus utilizes inside the host cell. And so there are other places where vaccines could target, and they have been ignored so far. So there's another way of circumventing the current uh, dilemma.
Okay. I appreciate that answer, sir. The next question is by Srivatsan. Srivatsan asks, can you tell something about cryo-TEM and cryo-ET, which were used to observe tunneling nanotubes recently?
Um, what was the question again?
Uh, oh yeah, I'll repeat it. Can you tell something about cryo-TEM?
TEM,
Yeah, and cryo-ET.
cryo-ET.
Yeah, which were used to observe tunneling nanotubes recently.
Uh, well, TEM is EM. Okay, all the
All right.
microscopes that we use are
All right.
transmission microscopes.
Okay.
And cryo-ET is ET stands for electron tomography, and this is when I showed my mentor's idea of tilting one uh, uh, specimen into many different angles. That's cryo ET. Okay, if
All
you
right.
do this in cryo, uh, and my lecture was on uh, uh, cryo-TEM of single particles, okay, which is different from cryo-ET of other kinds of specimen. Okay, so when you cannot when you cannot bring your sample into the form of individual identically structured particles, and you are forced to use to look at things in their original context. Then it's
Right.
called cryo ET.
All right. Okay. Um, could you tell us a bit about the Frank Lab? What are the major research activities you and your team carry out there?
Um, well, uh, I told you before that the now after 2017, I'm
Mhm.
sort of free to go into areas that are less explored and more high risk. And
Okay.
so two of these areas are one of them is time resolved cryo. So we want to know something about the course of reactions. We put two components together, and then we want to see how this reaction develops. And we want to trap short-lived states that might only live for 50 milliseconds or 100 milliseconds or so. And so we have to trap these. And so I'm using microfluidic devices to to define the time that the reaction can occur. So these kinds of experiments are not possible with a standard technique because it takes too long to put a droplet onto onto a grid and then blot and then plunge. This is all that takes at least a few seconds.
Right.
And so so I'm working on this, and we have two publications in Nature Communication in 2019. You can look them up. These were the first biological results that have been obtained with that. And the other technique is taking advantage of the fact that we see hundreds of thousands of the same molecules in the thermal environment. So they are actually they're changing their conformation, and they assume all the different conformations that are possibly important for their function. And so these are now statistical techniques in order to map these conformational changes, which are in a continuum, and account for them. So these are mathematical techniques which can give us information on the energy landscape of a molecule.
Right, that sounds really interesting, sir. When was your scientific interest first sparked, and when did you develop an interest in electron microscopy?
My first scientific interest, I think, I'm tracing this back when I was 8 years old. I performed experiments underneath the veranda of my house, and so I think essentially I think I could later say that I did science before I knew what science was, and then later on. How I got into electron microscopy was simply by when I did my master thesis at the university in Munich. I had a mentor who was an early pioneer in electron microscopy, and so I developed an interest in the electron microscope. It's a very ancient electron microscope that he had in his office. So then after I finished my master thesis, which was not on electron microscopy, but involved electrons already, I looked for a lab that was active in electron microscopy, and that's how I found Walter Hoppe in Munich.
Okay, that's really interesting. So to finally end the question and answer session, do you have any message for all the students watching this live lecture right now?
Well, I told you about the importance of peripheral vision, and then I think it's also important that you have to scrutinize everything that other people tell you, including your mentor. Okay,
right.
Mentor can be wrong, and in my case, it was a case where my mentor was not on the right track, and so you can develop your ideas not just under direction of somebody, but even in contradiction of somebody.
All right, I'm very sure all the students really appreciate you saying that. So with this, we have come to the end of today's lecture and Q&A session. Thank you very much, sir, for those truly wonderful and informative answers. Once again, thank you for joining us for our Spotlight 2021, especially considering the time difference between India and the US.
Yeah, it's just starting to get bright here.
Yeah. We also thank the audience as well for tuning in. and to watch this delightful lecture. Please find the attendance and feedback form link on the chats. Kindly do fill the same. Participants who attend 4 or more live lectures as a part of Shaastra Spotlight 2021 would get e-certificates. Tomorrow on Shastra Spotlight, we have 4 amazing fireside chats lined up for everybody. At 11 a.m. IST, catch our fireside chat with the former chairman of ISRO, Dr. K. Radhakrishnan, followed by a fireside chat with the godfather of Silicon Valley, Dr. John L. Hennessy, at 1 p.m. IST. At 3:30 p.m. IST, we will have Nobel Peace Laureate Dr. Mohammed Yunus with us, and finally at 5 p.m., Dr. Viral Shah, CEO and co-founder of Julia and the author of Rebooting India. We thank the audience for your patience and hope you had a wonderful time. Thank you.